Hello, Dan Guerra here for another session of VerEvMed, where we are going to discuss today a client's interest in Gaucher's disease. So let's go ahead and get started right away. So Gaucher's disease, which is also known as a lipidoses, a specific lysosomal lipid storage disease, has a specific lesion in the glucocerebrosidase enzymatic activity. We'll take a look at the reaction that is deficient in this particular disease. But I want you to understand that a glucocerebrosidase, for context of Gaucher's, is really a D-glucosylceramidase enzyme. So we're going to be removing the glucose residue from a glucosylceramide lipid, which is normally found in the membrane. Okay, these sphingolipids are really important not only for membrane integrity, that is structure-function relationships, but also as signaling molecules. They are found in lipid rafts as they move in from the plasma lemma back into the cytosol via endosomal rafts, lipid rafts. And in that process, they also carry cargo. What kind of cargo? The kind of cargo that is including sphingolipid, phosphoglycerolipid, as well as a host of proteins that make it to that plasma membrane. Proteins such as signal transducers, G proteins, as well as a host of receptors, including the insulin receptor of note. So remember that I'm coming from VerEvMed. My name is Dr. Daniel Guerra. And what we are scientists, and what we do for our business, we verify published evidence in medical biosciences. That's what we do. So again, this is a client-based question asking about Gaucher's disease. So in order for you to understand what the enzymatic lesion is in the disease, you have to know a little bit about the substrate for that enzyme. So let's take a look here at these basic structures for a few minutes, if you will. There are two basic lipids that we can talk about in sphingolipids. There's also one just simply called sphingomyelin, which you find associated with the neural tracts. Okay, so you have astrocytes and you have specific bundles of lipids that are around these axons, and these particular lipids help in signal transduction in the central nervous system. That's what we're talking about here. We're talking about lipids that are involved intimately in the nervous system, both in the central and the peripheral. So what are cerebrosides, as you might guess, are found in the brain. They're the simplest of the sphingosine-based carbohydrate-containing lipids, and they usually just have like a glucose or a galactose as a carbohydrate moiety. The gangliosides are far more complicated. They contain several different kinds of carbohydrates in various linkages, beta-1,3, beta-1,4, alpha-1,3, linkages like that, glucosidic linkages. And you can have up to 7 monosaccharides per lipid moiety. So these are really complicated glycolipids. All right, so take a look down here. You can see that sphingosine is now your base here. Okay, so these are going to be sphingosine associated lipids, not glycerol. So sphingosine takes the place of glycerol in sphingolipids. And we'll talk a little bit about how you make sphingosine because it's an interesting biosynthetic pathway. At any rate, in cerebrosides, you see you got this monosaccharide, glycosidic linkage there. Then you've got another unusual feature of sphingolipids, which is rather than having an acyl linkage, an oxygen ester, you have an amide linkage of that fatty acid directly to the sphingosine base. Okay, so it's linked to a nitrogen atom. So in order to remove that fatty acid, it's going to be a different kind of phospholipase activity than you're going to have with conventional phospholipases working on glycerol phospholipids. Okay, so right there, there's another unique characteristic. So those are the cerebrosides, and the gangliosides, as I said, just have an oligosaccharide here in this glycosidic linkage. That's a major difference. Okay. In the biosynthetic pathway, you have ceramide here, and I want to just point out a little bit of the structural features of this really interesting lipid. I find it to be really a cool lipid. It's quite hydrophobic, and ceramide itself has a lot of interesting biological aspects and features. Ceramide itself is an important signaling molecule, and I talked about it in one of my previous VerEvMed lectures when I was talking about apoptosis. So ceramide is associated with programmed cell death by inducing a host of cascade systems, including caspases and mitochondrial degeneration that ultimately lead to cell suicide. Ceramide doesn't just do that, but that's one of its major known features in the cell. But ceramide is made up of palmitate, so that's palmitic acid with an unusual trans bond in it, and serine, L-serine. So it is a condensation product of L-serine and palmitic acid from de novo fatty acid synthesis or from the diet. Then there's your nitrogen-linked fatty acid there. Now, the kind of fatty acid you find there is usually a very long chain, either a monounsaturated 20-carbon fatty acid, or sometimes we find those polyunsaturated fatty acids with multiple double bonds, either omega-3 or omega-6. So these are complex lipids, and they have a lot of signaling molecules associated with them upon hydrolysis and further metabolism. So when you start off with ceramide as your base structure, already it's a complicated lipid. The way that you make sphingomyelin, which again is in the sphingomyelin sheath, right, around axons in the central nervous system, is all you have to do is pick up phosphatidylcholine from phosphatidylcholine, that is the glycerolipid. Product known as diacylglycerol and sphingomyelin. So there's phosphatidylcholine. That's the same quote-unquote polar head group you have on phosphatidylcholine, only now it's on a sphingolipid, and we call this sphingomyelin. You can take that same ceramide backbone, and now you add an activated sugar. Here we're just using UDP glucose. Obviously, this could be galactose or any other, usually hexose sugar. And nucleotide carbohydrate metabolism is most often the way you activate these sugars, right? So you run it through the lower pathway, and you make these nucleotide sugars, and then you add covalent addition now of these sugars. In this particular case, you made the cerebroside. So there's glucose associated here, as we saw a few slides back. That's the actual structure. You still have the amide-linked fatty acid, and of course you have the sphingosine base. Now, this serves as precursor to add more sugars, and that's where you make the more complicated gangliosides, and there's a whole host of those structures. So here's just your standard straight-up model. There's sphingomyelin, and there is glucose cerebroside, right? There's glucose there. You can see there's an N-linked fatty acid, and again, this is palmitic acid with that unusual trans double bond. Now, that's biosynthesis. I didn't actually show you how ceramide was made, but I explained how you make a sphingosine base. The reaction that is affected in Gaucher's disease is this one here, the glucose cerebrosidase, right? It's the one that's going to remove ceramide. If you start off with the glucosyl ceramide, right, also known as a glucose cerebroside, right? It's just ceramide gives you more specificity of what you're talking about. You're going to make ceramide, and you're going to make glucose. Okay, plain and simple. That's the reaction that's deficient in Gaucher's. You can also, however, take glucosphingosine. Okay, now notice the difference between this lipid and this lipid is you don't have this additional fatty acid here. You don't have this N-linked fatty acid, right? So, but you do have glucosylsphingosine in the membrane, and you do get this reaction. It's not often talked about in Gaucher's, but it is the same enzyme, and indeed a deficiency in that enzyme is not going to allow you to remove this sphingosine from glucose or ceramide from glucose. Now, the big deal here about this is that this is a lipid storage disease, also known as a lipidosis, or lipidoses in the plural. Now, why is that significant to the cell? Well, these lipids need to turn over. In other words, plasma membrane-associated lipids need to turn over. They need to be back endocytosed into the cytoplasm via endosomal trafficking to the lysosome, where this reaction takes place. And then those lipids are broken down and then resynthesized. That takes not only that endosomal trafficking compartmentalization process from the plasma membrane and the lysosomal machinery, all the enzymes and all the other membrane-associated fusion reactions that are necessary. But you also have, in the rebuilding of these lipids, the mitochondria, the endoplasmic reticulum, the peroxisome, all working together, orchestrated to make these sphingolipids. So it's quite a complex process to synthesize them, to retailor them, to put double bonds into those fatty acids, and then to bring them back down. that whole orchestrated process has to turn over. When it doesn't happen, you get these severe diseases. They're often targeted, neural based, as we'll see Gaucher's can be, at least with type 2 and type 3. Okay, so these are very devastating diseases. They're inborn errors of metabolism. That is, they are genetic. They're usually carried on as autosomal recessives, as we'll see in a moment. So the take-home message here is that the reaction is actually deficient or absent in Gaucher's is going to be this glucocerebrosidase, right? That reaction right there. So as I've been hinting, there are 3 types of Gaucher's. Type 1 is the non-neuropathic, also known as chronic. It occurs at any age with a variable severity for the patient. Clinical signs include hepatosplenomegaly, pancytopenia, osteopenic and osteolytic degeneration of the skeleton. And this disease, type 1, tends to be pan-ethnic. That is, all ethnic groups get this disease, but there's a slightly higher frequency of it in Eastern European Jews. Type 2 Gaucher's disease, far more severe. This is known as acute neuropathic. Okay? It's rapidly progressive once it comes on. It happens to very young children, infants, in fact. It is pan-ethnic. It is a neurovisceral lipid storage disease by trade, and it results in early death. Again, there is no known, except if we're talking about gene therapy, which has not really been frequented here for this disease. There isn't any cure. There aren't any drugs for these diseases. Right? We know a lot about them. We've known a lot about them since Gaucher first described them back in the 19th century, for this one in particular. But we just do not have the tools to be able to do anything to really preserve people that have particularly type 2 and type 3 disease. That's why people study it. We're trying to look at different elements of this disease, not just that one enzymatic reaction, find out what's the downstream effect of that, and find out whether or not we can build back up maybe some of the products that are normally impacted by that loss of the glucosyl cerebrosidase, and maybe be able to ameliorate this disease in those patients. Type 3 is known as subacute neuropathic. It's less aggressive than type 2, but basically it's the same kind of disease. Some of the other presentations include ataxia, myoclonus, seizures, dementia also develop in this disease, of course. You get some hepatosplenomegaly. You also get death in early childhood. Usually these kids can make it at least to puberty, but that's not always the case either. Again, it's a pan-ethnic disease. They all are sort of pan-ethnic. However, this one has a rare genetic subisolate, which you find in northern Swedes. Okay, so there is some element of a specific ethnic group for type 1 and type 3. But for the most part, these are pan-ethnic diseases, lipid storage diseases. So as I've been saying, all Gaucher's is autosomal recessive. You need both mutations for this to be a problem. So if you have one copy of the gene and it is expressed correctly, you're not going to have this disease. So you have to have both of them disrupted. And again, what happens, the ultimate biochemical consequence of the loss of that enzyme is the glucosylceramide accumulates. And then where does it accumulate? In reticuloendothelial lysosomes. When it starts showing up in those lysosomes is when you start getting all of those presentations downstream, right? So how does that happen? Well, one general global effect is that because you don't have good breakdown of those lipids, and because you have a buildup of those lipids in the endosomal compartments, you change the entire architecture of that cell. So the plasma membrane no longer has the appropriate retailored lipid moieties. And not only that, the proteins that get normally rafted through that lipid rafting process using the ER and the Golgi to the plasma membrane are not packaged correctly. So you get a lot of the proteins that normally act as receptors on the surface of those endothelial cells that are no longer there, no longer packaged appropriately because you have a problem with the breakdown that is the turnover of this lipid. So this is a major, colossal devastation to the cell. Whenever you do anything with the endosomal compartment and you alter lipid trafficking, you're going to end up with a very sick tissue. And that's exactly what you see here. That's why these are so devastating and so lethal, these diseases. So here's your standard phenotype. Here you're looking at the enzyme activity In the Gauchers, you see that's very low compared to a healthy control. Okay, that's about what, I don't know, maybe 10 times less. Now, what else is interesting about this disease? A consequence of having the low levels of the glucosidase, of the cerebrosidase reaction, is you get a turning on of a chitotriosidase. Now, this is an unusual enzyme that normally is not overly expressed. You always have some of it, however. Now, this is actually a defensin type of protein because chitin, which is made up of N-acetylglucosamine repeating units, is a normal cell wall product in fungi. It's not the kind of compound the cell normally sees, the mammalian cell certainly doesn't see very much of, but you have a constitutive activity of this enzyme. However, because you're building up these complex lipids in these endosomal compartments, that turns on the machinery that induces the expression of this chitinase type of enzyme, because the cell recognizes that undegraded lipid moiety in the lysosomal endosomal compartment as something looking like chitin. So it really jacks up the activity of this enzyme. There's 16,000 nanomole per hour per milliliter for in the Gaucher's disease, down to only 50 basal activity in the healthy controls. You see a tremendous thing. So I want to tell you a little bit about this chitotriosidase, because it's something that's not often thought about or looked at in Gaucher's, but take a look at this. So we're going to call this enzyme just CHIT1. It is a chitinase, and it is massively expressed in lipid-laden tissue, and you see it in macrophages in Gaucher's disease patients. Its enzymatic activity is markedly elevated in the blood of patients suffering from a whole host of lysosomal lipid disorders. So it's a general defensin type of enzyme, as I mentioned. So you see it in sarcoidosis, thalassemia, as well as visceral leishmaniasis. Okay, so various kinds of parasitic infections and pathogenic determinants can induce the increase in CHIT1. What does the enzyme actually do? Very interesting, actually. Not only is it involved normally in the breakdown of chitin, and that's not really the problem here, it has other effects. In fact, it induces a phosphorylation of the TGF-beta pathway. Okay, this is going to be a pro-inflammatory cytokine pathway binding to its receptor. When you phosphorylate that receptor system, it acts as a tyrosine kinase, and it turns on the SMADS complex. When it does that, you phosphorylate SMAD2 and SMAD3. That forms a dimer. SMAD4 is picked up. Then you make this trimer of these three SMAD proteins. Those then act as a transcription factor. And what do they do when they get to the nucleus? They turn on the production of pro-fibrotic mediators, collagens, and other ECM proteins. Okay, so extracytoplasmic matrix protein synthesis is what happens. The overall global take-home effect is this is an increase in fibrosis. Okay, so this is one of the issues that you see in all these various other kinds of disorders, right? That's why CHIT1 is such a problem, because it actually turns on transcription factor vis-a-vis or through the TGF-beta. So here's a paper I found in the Journal of Epidemiological Pharmacology. I just took a hunk of the abstracts rather than me cutting it up and just give you an idea. First of all, this is what chitin looks like. I told you it was repeating units of N-acetylglucosamine. There it is, right? Just beta-1,4 linkages, nothing fancy here. Here's from the abstract. Let's take a look at this. Mammalian chitinases, right, belong to a glucosyl hydrolase family based on structural homology and family. So it's a large family. You find this in bacterial and in the eukaryotic mammalian chitinases. Among the mammalian ones, you have this CHIT1, as we've been talking about, which is greatly elevated in Gaucher's. And you have an acidic mammalian chitinase called AMCase. Both of these can be elevated in Gaucher's disease and other lipidoses. They're capable of hydrolyzing that beta-1,4 bond. And when they do, they're going to cause, again, that buildup, right? They're going to cause that buildup of fibrosis. So that's what they're not doing that because they're breaking down this bond. They're doing that because they're turning on the TGF-beta. Okay. Now take a look at this. CHIT1 plays a pivotal role in the context of infectious disease, including malaria, fungal diseases, of course, and fungal infections, because again, this is a chitinase that's turned on by that fungal infection. and it's a host defense toward chitin itself and the pathogen cell structure. Diagnostic marker of the disease, in fact, when you get a fungal disease. Now, in contrast, KIT1 is released by activated Kupffer cells, okay? And of course, it's in the liver, and they can induce hepatic fibrosis. We just saw how that happens by turning on the biosynthetic machinery at the transcription level for those ECM proteins, right? You see it in hepatic fibrosis. You also see it in cirrhosis, alcoholic cirrhosis. You get increased serum levels, as we saw that whopping increase in SHIT1. And you get it in Gaucher's, you get it in bronchial asthma, you get it in atherosclerosis, and we also saw those other disorders where it's also common. All right, so that's enough about that chitinase. Just want to bring that up because it's normally not described in Gaucher's, but it's there. Possibly a potential pharmacological target, right? Because it's definitely inducing some of the downstream processing of this disease and probably some of this presentation. Now, here's this paper I want to look at. Okay, now this paper here, we're looking at, back at the beginning, I told you we're going to be looking at a paper published in Clinical Biochemistry. If I didn't, that's what we're looking at here. Citations at the beginning, 2017 this was published. Now, what they show in this paper is that in Gaucher disease, besides the fact, or also, of course, the fact that you've got this great decrease in cerebrosidase activity, basically negligent activity, you also get less BDNF. What's BDNF? Brain-derived neurotrophic factor. You get a greatly reduced level, even though there's a lot of variation around the standard deviation compared to controls, okay? And it's tightly packed, so you can see there's just an overall decrease in BDNF. Why is that important? BDNF is really important for neuronal maintenance, okay? BDNF is important when you have neural damage, neural tissue turnover. Low levels of BDNF, which is basically a growth factor for central nervous system, does mean that repair of those neurons is greatly diminished. Okay, now you don't want an overactive BDNF expression because then you might be talking about glioblastoma, but you certainly don't want to be at this very low basal level because apparently that's associated with some of the CNS effects of Gaucher's. Okay, all right. Now that's interesting in itself. Now take a look at this. With tumor necrosis factor, you also get an increase in TNF. Okay, that's really significant as well. You get an increase in TNF. That is, of course, what? A pro-inflammatory cytokine as compared to controls. You also get an increase in another very potent pro-inflammatory cytokine, interleukin-4 in Gaucher, right, as compared to healthy controls. So you have TNF alpha and you have interleukin-4 both elevated as BDNF is decreased in Gaucher's. Very significant effects at the immunological level and also at the level of neurotoxicity, right? All right, now this is an interesting graph. Here we're looking at Gaucher's, the black squares, and control is the white squares. And you can see here that now we're looking at interleukin-4, a lot of interleukin-4. We just saw that in the Gaucher patients. We see a greatly reduced amount of BDNF. Now, is this a causal link or a correlated link? We don't know, but we do know that they are linked. You get less of the brain-derived neurotrophic factors, see way down here, and you also get an increase in interleukin-4. So there might be an immunological aspect to this suppression of BDNF4. Likewise, the same thing when you're looking at tumor necrosis factor. You're all clustering here, right? You're getting more of that pro-inflammatory cytokine. You're also getting a whole lot less of the BDNF as compared to your healthy controls out here. Okay. So, again, here's the paper, Clinical Biochemistry. It was published just last month. I'm giving this talk right now to you, this video, in April, mid-April of 2017. So this paper's a month old. This is not open access paper, so you'd have to have a subscription to it or have access to a library to be able to read the whole paper, but that's what I'm doing for you here. Now, let's take a look at some more of the summary of this, because it's a very short talk today. Besides the accumulation of glucosylceramide, which is what happens with type 1 Gaucher's pathology, okay, there is, according to this paper now, a new report, a decrease in BDNF, okay, which again is going to decrease the amount of neural growth, right? Synaptic connections are going to suffer when you have lower levels of BDNF. You're also getting this increased level of pro-inflammatory cytokines, TNF alpha and interleukin-4. Now, one other element here, which is very interesting, which this paper talks about. And I'm not going to go into detail here because I've been promising for a while I'm going to do a nice lecture on epigenetics. But another thing we see in Gaucher type 1 is a hypoacetylation of histone 4. Now, I've talked about this in a few other lectures, and I'm not going to go through all the details of it, but of course, cohering histones associated with chromatin in the nucleus, those histones, when they become acetylated, that opens up the chromatin both for replication and for nascent transcription. When you have hypoacetylation of the histone, that's going to diminish the amount of gene expression, okay? So it's going to be a more quiescent chromatin structure when you have hypoacetylation. And indeed, it appears that the Gaucher phenotype, endophenotype, is a decrease in the acetylation of the lysine residues on histone 4 that generally then earmarks less level of global gene expression in those nuclei. So you got 3 different possible new ideas for pharmacotherapies, right? You're looking at maybe elevating BDNF, and there are ways to do that. There are drugs out there that have been targeting BDNF for a long time because of the various toxicities that induce that, such as hypoxia and hypoxic shock in the brain decreases BDNF, so we know for a long time we want to elevate that. Lower levels of BDNF are also associated with late-stage Alzheimer's disease and Parkinson's disease and prefrontal dementia. Elevation of pro-inflammatory cytokines in the central nervous system is also not a good thing in general. So is there a way to target maybe the receptors for those in the brain, or maybe decrease the amount of circulating TNF-alpha or IL-4 that makes it to past the blood-brain barrier, or that is associated with either microglia or neurons, frank neurons in the central nervous system. Likewise, there are a whole host of drugs working on sirtuins, and sirtuins are deacetylases. Again, I talked about those enzymes. There's multiple forms of those. So a deacetylase would then give as a product hypoacetylation. So we're wondering here, is this accumulation of glucoceramide because of the deficiency in glucosaraminidase inducing perhaps sirtuins, giving you this hypoacetylated pattern? We don't know, but obviously that's another potential for targeting for pharmacotherapy. So that's all we're going to talk about today. Just again, that brief discussion of Gaucher's disease. Again, I'm Dr. Dan Guerra. There's my email address, which you can contact us directly. There's our website for VerEvMed. And again, what we do for a living is we are scientists verifying published evidence in medical biosciences. Today we talked about a client's interest in Gaucher's disease. We could be talking about your subject matter next time. So thank you very much for your attention, and please have a wonderful day. Goodbye.